Yes.
Okay, the supervisor is Dr. Asmida Ismail. And please be reminded that you have 8 minutes for presentation and 2 minutes Q&A.
Okay.
Okay.
Can I start now? Assalamualaikum and a very good afternoon to our respected panels and my fellow classmates. My name is Aina Shafika binti Muhammad Syahudi, and today I will be presenting an article review with the title A Review on the Morphology and Taxonomy of Red Algae Rhodophyta in Malaysia. I will be presenting the article review in 6 parts. First and foremost, the introduction. Algae belongs to a group of photoeutrophic, multicellular, non-vascular plants due to the lack of stem, real roots, and leaf. Microalgae are classified into 3 major groups, which is
Aina,
Phyllophyta.
Dr. Siti Khaira boleh mute tak? Aina, you punya
Boleh
slide
tak?
tak bergerak.
Oh, slide tak bergerak.
I think you kena share, boleh buat PowerPoint mode.
Okey dah ni. Ni okey kot.
Tapi I nampak kecil ke ni through phone
Oh, sekejap.
Dia kena buat PowerPoint mode.
Tak nampak besar.
Ah,
Oh, saya rasa saya kena left sekejap sebab ada problem. Sekejap ya, saya tengah share. Dah nampak ke?
tak keluar lagi.
Tak keluar lagi. Macam mana ya?
Ini you share entire screen ke apa? And then you tekan slide show tu yang bawah.
Tekan slide show.
Bawah kanan ada slide show mode kan? And
Saya dah tekan.
I tak tahu yang lain nampak macam mana ya. I nampak sangat kecil.
Ya, kecil lah.
Dia kecil.
Kan dia
Tak,
macam
dia tak apa kalau mood yang ini, tapi kenapa dia macam
dekat
phone lah. Kenapa dia
macam kat phone.
bukan melebar?
Oh.
Awak tak besarkan screen, maximizekan screen atas ke? Itu okey tak? Yang tadi atas tadi tu. Yang sebelah kanan atas sebelah X tu. Oh awak tu.
enlarge.
Okey.
Baru okey. Lepas tu slide show. Ni
Awak
dah slide.
okey, sekejap faham. Aina semua nak kecilkan sikit sebab ada skrip kat sebelah
So kena kecil.
tak je. Sebab kalau ada skrip kat sebelah boleh guna yang share window instead of entire screen. Tapi tak apalah kalau tak ada okey je macam ni. Macam ni okey macam ni.
Kalau macam ni je okey ke?
Okey lah. Cuma
Okay,
I
untuk saya
okey lah.
Sebab kalau kita buat show screen biasanya kadang tak bergerak. So kalau macam ni usually dia bergerak. Cuba buat gerakkan Syafiqah Aina.
Sekejap
Pergi 3, pergi 4.
Tapi thumbnail yang sebelah tu tak boleh padam tak boleh kecilkan dia supaya nampak view content tu je besar ke?
Yang sebelah kiri tu punya.
Yang sebelah kiri tu okey jap.
Kecilkan dia punya apa nama?
Sebelah kiri tu padam
You gerakkan dia punya macam tak tahulah divider ke apa nama dia tu.
Tak boleh.
Bukan Aina. Yang sebelah kiri yang awak punya thumbnail tu yang kecil-kecil ni, sebelah kiri tu. Kecilkan dekat situ. Maksudnya awak ke kiri kan lagi? Okey macam tu. Pergi kiri sikit.
Yes. Okay lah. Better dari tadi tu. Cuba next.
Next.
Okay.
Awak zoom tu, awak kurangkan zoom dia. Zoom out sikit.
Okay, boleh mula kot.
Okay. Saya minta maaf. Masalah. Saya start sekarang ya.
Okay.
Okay, Assalamualaikum and a very good afternoon to our respected panels and my fellow classmates. My name is Aina Syafiqah binti Muhammad Syahudi, and today I will be presenting an article review with the title A Review on the Morphology and Taxonomy of Red Algae Rhodophyta in Malaysia. We're presenting the article review in 6 parts. First and foremost, the introduction. Algae belongs to a group of photoeutrophic, multicellular, non-vascular plants due to the lack of stem, rear roots and leaf. Microalgae are classified into 3 major groups, which is Phaeophyta, Chlorophyta, and the main type of algae population, which is Rhodophyta. There are more than 6,200 species currently documented in the red algae, but with new species defined each year, the taxonomy is rising. The aim of this article was to list and describe the red algae by collecting both previously described and potentially unknown taxa from related articles, and to collect and record the taxonomy of Rhodophyta species in Malaysia and produce a checklist of Malaysian red algae in general. Moving on to the body of research, Rhodophyta is known for its morphologically and taxonomically varied lineage of photosynthetic protists. Rhodophyta comprise of 2 subphyla and 7 classes. Not to mention, Malaysia is one of the most researched nations for the variety of Rhodophyta species. Since the last checklist was released, Malaysia's seaweed list has greatly increased due to the increased number of algal taxonomies in the nation, regional cooperation, and the efficient use of scientific device to distinguish effectively between taxa that are similar in their morphological condition. The research questions that can be identified in the article were how Rhodophyta species varies from each other, how to determine morphological characteristic of Rhodophyta species, how to assess taxonomical group of Rhodophyta species, how morphological characteristics influence in taxonomical of Rhodophyta species, and what is the conclusion from this review. Following that, results were obtained by extensive research from related articles. Currently, Malaysian red algae sample have risen up to a total of 28 species. In genus Batrachospermum, 3 species were identified in Malaysia. 2 of them were Batrachospermum cylindrocellular and Batrachospermum berriense. Batrachospermum cylindrocellular was identified and collected from Tasik Berak in Malaysia, and this species is different from other species by having cylindrical fascicle cells and differentiated carpogonia branches and the presence of spherical carposporophyte in the outer part of the wall. Batrachospermum berriense was also discovered in Tasik Berak. They have long carpogonial branches and short carpogonia. This species commonly mistaken as other Batrachospermum species because they have the same whorl appearance. In terms of taxonomy, this genus is made up of 9 sections. Batrachospermum cylindrocellular initially was placed in Batrachospermum section. In-depth studies suggest this species is related to Aristata section. Batrachospermum berriense was also identified as a member of Aristata section. Eucheuma alvarezi was obtained in southern Carindingan Island Semporna, Sabah, Malaysia. This species distinct from other Eucheuma species because it has multi-axial teli, a simple discoid holdfast, and a primary branches. The branches are uneven and unilateral, and this species has no or very few natural second branches. The special characteristic of this species were the bending and the length tapering of its extremely smooth main branch. In other Eucheuma species, secondary branches grow between or after the presence of primary branches, but in this particular species, there are usually no secondary branches. So here is the example of Eucheuma alvarezi, Telly, and Holdfast. A total of 3 Rhodophyta species from genus Glacilaria have been found in Sarawak, Malaysia, which are Glacilaria blogeti, Glacilaria chengi, and Glacilaria coronopifolia. Glacilaria blogeti was identified as dark red in color. Thallus might reach up to 200 mm in length, cylindrical branch, and the holdfast was semicircular in shape. These species also look compact due to the unique formation of secondary branches. Glacilaria chengi are also dark red in color. Its thallus could develop to 220 mm tall, and both primary and secondary branches can grow up to 40 mm and 170 mm respectively. Glacilaria coronopifolia had a combination of red and purplish in color, and also have cylindrical discoid branches. At the end of the thallus, typically new short branches with pointed tip were created. And here are the visual example of Glacilaria blogeti, Glacilaria chengi, and Glacilaria coronopifolia. Morphological observation had been done for Hymenoptera malaysiana in Pulau Besar Melaka and Pulau Karindingan Sabah. These species have flexible cartilaginous folios, red brownish pink belly with slimy texture. Cystocarpic and tetrasporic telly for this particular species were identified and examined. This species is distinguished by a telly which possesses oblong blades with a soft cartilage structure and slender texture, resulting in a tiny staples discoid holdfast. Here are the visual example of thallus and blades from Hymenoptera malaysiana. In conclusion, Malaysia is rich in flora and fauna biodiversity, not to mention about the Rhodophyta species can be found throughout the entire country. For example, Betracosperma species was found in Tasik Berapahang, Eucheuma species and Hymenoptera species can be found in Karindingan Island Sabah. Meanwhile, Glacilaria species were identified mostly in Sarawak but can also be found in Penang, Melaka, Negeri Sembilan, Selangor, Kedah and Johor. I would suggest researchers to learn with much detail about the local climate and the species adaptation on the local weather before conducting any observation. This due to the fact that some Rhodophyta species are seasonal. Last but not least, here are the references that I used for this article review. Okay, thank you. That's all from me.
Okay, thank you, Aina Shafika. Any question from the panel? Okay, Aina, you are doing the article review on different types of red algae, right?
Yes.
So based on your reading and also literature review, can you suggest any species of interest from your reading? Any species of interest that has potential to be studied in future in terms of their distribution, or maybe because of the rarity of that species? Can you share your opinion on that?
I think from my observation, I think Glacilaria had the most potential for a future observation because they have many Glacilaria species can be found in many places, and also they have many types that can be observed. We can observe them based on colors, based on based on color structure and their adaptation to the environment.
So only that Glacyracea species.
Yes, because
So
for other species, then they are not much in Malaysia.
okay, so that species is widely distributed in Peninsular Malaysia and also Sabah
Um,
Sarawak, or just in Peninsular Malaysia?
both Peninsula and Sabah Sarawak.
Okay. So other panel, perhaps you have one more question.
Yeah, I know.
It's okay. Okay, Siti.
Okay.
That's okay.
I know. May I know what's the differences between red algae and other algae? Short answer, please.
Red algae is based on the name. Commonly, the algae is red in color.
Kurang tepat lah tapi masa dah habis lah.
I'm sorry, saya tak boleh dengar.
Okay, I think we have had enough 2 minutes, yeah? 2 minutes for Q&A. So thank you so much, Aina Shafiqah.
Dr. Faiza,
Yeah.
can I ask simple question?
Okay.
Okay, Aina, from your
Yes.
research questions, question that you listed before, after you did this study, is it all the research questions has been answered?
Yes, all the research questions have been answered.
Okay. So please put in your conclusion, yeah?
Okay.
Every research question been answered. And one more thing is you are using the wrong logo. I think UiTM logo. You should use the formal UiTM logo, the official one.
Okay.
This one
Bye,
is not official logo of UiTM. Okay, thank you.
Thank you, Dr. Siti Asma and Aina Shafiqah. So next, can we have Nur Shasha?
Yes, betul.
Mana dia, Nur Shasha?
Sekejap tengah present the share screen.
Oh, okay. Okay, dah ke? Dah ready?
Okay.
Okay. Okay, boleh mula?
Sorry, I'm waiting for the bell to be.
Oh, Bell, Dr. Siti Khairia.
Saya dah ring tadi lambat sangat.
Oh, tak dengar.
Okay, I start now. Assalamualaikum. I speak to all the panels present here and to my fellow friends. My name is Nur Shasha binti Abdul Rahim. I am from AS201 683. I am under the supervision of TS Dr. Judith Kisip. So this is an overview of my slide presentation. For the introduction, this study mainly focused on the shrubs community that live on the ground floor area in the Pasoh Forest Reserve, Negeri Sembilan, to look into by looking into their species diversity, abundance, and also their species richness. The findings of this study can contribute to the understanding of the forest dynamic, especially in terms of the forest regeneration in the study area. In this situation, it may lead to a deeper understanding of the shrubs of the Pasoh Forest. So the objective of the study to determine the diversity and composition of shrubs vegetation and to quantify the shrub species that are abundant based on their species and their taxonomic composition. So collecting data on the ground was not possible due to some restriction on from the ongoing pandemic of 19. The secondary data was obtained from Pasoh Research Secretariat Committee, The data was obtained using a quadrat sampling method. It is the most effective method to approximate the total population in the forest. Paleontological statistics software is used to analyze the data based on three factors, which are species diversity, richness, and evenness. Shannon Diversity Index and Markgraf Richness Index formula were used to assess the diversity of species in its population. However, in this study, there is no specific measurement of diameter breast height due to the data that was obtained is lacked in the column of the DPH. Thus, this study mainly focused on the overall random species of the shrubs, regardless of their sizes. Next, this is the result of this study. There are about total of 309 plant individuals that are present in the 25 sampling in Pasoh Forest. As you can see in the table, Rubiaceae is the largest family represented with 12 species in 7 genera. This is all the shrubs variations found in the Pasoh Forest. Only 3 individuals can be distinguished based on their genus, which is Ardisia sp2, Leucanthus sp1, and Pavetta sp1. The most abundant species is Rosmania macrophylla with 18.77 of relative abundance, followed by Ardisia sp2 and Lepicenthis tetraphylla with 12.62 and 11.97 respectively. Okay. The density of ground floor vegetation in the study plot was 309 individuals per hectare. Rosmania macrophylla again was the densest species with 58 individuals per hectare, followed by the Ardisia sp2 and Lepicenthis tetraphylla with 39 and 37. Rosmania macrophylla also recorded the highest frequency of 80%, which occurred in 20 out of 25 quadrat. Pavetta sp1 recorded the second highest frequency with 76, and Lepicenthis tetraphylla recorded the third highest species with 58 frequency of occurrence. So as for the description, the 3 plants, which are the Leucanthus sp1, Pavetta sp1, and Ardisia sp2, could only be distinguished by their genus, or maybe due to the limitation of identified of species. Anonaceae were among the least abundant family, or maybe due to their inability to see more than 1 centimeter DBH, making their density look very low. So, Pasoh Forest Reserve recorded the Shannon Winner Diversity Index of shrub with 2.63, which indicates a value below 3.5. This show the forest is relatively poor in diversity, and Shannon Evenness Index with 0.5137. As for the Markgraf Richness Index, it shows a low index with 4.535. The fact that it is dominated by the single species, which the Rosmania macrophylla, it is not possible for the species to present in most quadrants. So this species accounted for almost 80% of the total number of trees in the plot. So in this study, Shannon Evenness Index has a moderate value when it varies near value 1 or not 0. So abundance of these 3 species was higher as the top 3 leading species, which is not possible for them to present in most quadrat. This study can be proved by the study made by Compartment et al. In 1990 that showed the trees are still the most frequent species that survive in Pasoh. The abundance of these species can be said that they are successfully adapted to the ecological habitat in the forest that allow the plant to make use of the resource entirely. For the species that have less than 20% frequency of occurrence, it may be uncommon due to their species entire population consists of just a few individuals or limited to a small geographic area. Other uncommon plants may be packed into a small area that is not included in the quadrat of interest. According to the group cited for the Encyclopedia Britannica, an area with a limited range of habitat is often species poor. Nevertheless, few species may be abundant due to lower competition for resources with other species. Next, overall, the shrub species in Pasoh Forest Reserve has a species diversity with Shannon Winner Diversity Index with 2.63 and Markgraf Richness Index with 4.5, as well as the Shannon Evenness Index with 0.5137. As the species abundance, Rosmania macrophylla that belongs to the family Rubiaceae recorded the most abundant, the highest frequency of occurrence, and the densest species of rock. Next, based on the finding of the study, are the several recommendations to consider. Firstly, to address the accurate species diversity, ecologists or scientists may need to identify the three genus with species name. The studies need to be done periodically to ensure the accuracy and stability of the data generated. Thus, more recent and new research should be conducted in Pasoh forests. And in addition, urgent care is required for species that are maybe at high danger of extinction to prevent additional reduction in their population size that maybe lead to the extinction of the species. Furthermore, long-term research should have a wider applicability because this study only focus on the shrubs community. Thus, more research on all sorts of tree species in Malaysia is needed. Lastly, here is my overall reference for my thesis. Thank you.
Okay, thank you, Dora Shasha Abdul Rahim. May I know your size of study area in Pasoh Forest Reserve?
20 meters square times 20 meters square for 1 hectare.
1 hectare. So, why do you think within that 1 hectare you can only enumerate like moderate? I can say moderate because saying poor density is like very harsh word to use. So why do you think that moderate species diversity obtained in that 1 hectare of study area?
Okay, thank you for the question. I think because this data actually, I got it from the data from the Secretariat Committee. The data has a lot of the alive and the dead plant species. So if we consider the alive plant also, it will be high density. But somehow when the plant is not longer alive, so it can be shown that the species can be decreasing.
So the secondary data obtained, it was actually how many years of work?
Actually,
1 year inventory, or maybe do you know that
I
specific?
think it is not. I think this is not the latest census because from the committee they said this is the from 2015 year in the year 2015.
Oh,
So
year
they
20.
rarely do the research on shrubs.
Okay. Okay, thank you. Anyone else? Any panel?
Hi, Shasha. May I know what range is for the Magalhae index to be high?
Can you repeat that, sir?
Because your Magalhae index is 4.5 and you said it's low, right?
Yeah.
What range is high?
For this question, I can't say because usually the research can be comparable, can compare with other studies. We can compare between other studies, and we can say that the research is the value is high or low. But in this study, I have not yet seen any research about. So I can say that this is very cool because I have seen other types of trees that recorded a high value of Magalhae with more than 35 trees. So I can make assumption from that. It's below from 5.
I think there's a range for Magalhae. It's better for you to get the actual number, yeah?
Okay, thank you, doctor.
Okay. Ada lagi ke? Tak adalah sebab dah lebih 2 minit ya. So thank you Nur Shasha Abdul Rahim. Okay, Dr. Siti Kara Singapura, yes. Next we have Muhammad Nur Akmal. Mana dia? Muhammad Nur Akmal. Okay. Oh, baru I cam muka you.
Sekejap
Oh, kita ada Prof Mansur ya. Hai Prof.
Hai, Assalamualaikum.
Waalaikumsalam.
Saya nak leave sekarang. Okay.
Oh, okay.
Terima kasih.
Sama.
Okay, good luck everybody.
Thank you.
Ready ke?
Boleh lah. Bismillahirrahmanirrahim. Assalamualaikum and a very good afternoon to our lecturers and my fellow friends. Ladies and gentlemen, through this presentation, I am Muhammad Nur Akmal bin Mamak here. Would like to talk about my article review entitled A Review on the Cultivations of Microalgae for Polyhydroxyalkanoates Production. Throughout my final year project, I was supervised by Professor Madhya Dr. Ismida Ismail. Nowadays, petroleum-based plastic consumption are expected to annually grow over the year due to increasing demands from the consumer. Continuous accumulations of petroleum-based plastic without control bring negative impact to the nature. Recycling has been acknowledged as an ineffective measure, saying it is impossible to cover up most of the plastic waste. In my review article, I have included a few research questions that will guide me through the process. Is there any alternative ways to replace petroleum-based plastic? What are the advantages of using microalgae for bioplastic production? What are the types of cultivation techniques can be used? How the steps of polyhydroxyalkanoates extraction being done? And does microalgae able to replace petroleum and terrestrial crops? Next, these are my objectives for the review article. To determine the finest alternative to replace petroleum for plastic production, to identify the type of cultivation techniques for microalgae, and to study the effect of basic requirement by microalgae on polyhydroxyalkanoates production. There are various alternatives that are available to replace the usage of petroleum with other biodegradable materials such as terrestrial crops and microalgae. However, terrestrial crops require suitable plot of land, suitable weather or season, fertilizer and pesticide, and require a lot of time to grow before it can be harvested for plastic production. In addition, using terrestrial crops as a raw source of bioplastic would be a limiting factor for human consumption. Moving on to microalgae. Microalgae has a rapid growth rate, broad environmental tolerance, since they can utilize the sewage and wastewater as its primary nutrient. Under environmental stress, microalgae can produce bacterial polyester known as polyhydroxyalkanoates, PHA. One of the common monomers of PHA is hydroxybutyrate that can form a short chain of length polymer forming a polyhydroxybutyrate, which is known as PHP. In this review article, I have included the cultivations of microalgae consists of three types, which are autotrophic, heterotrophic, and mesotrophic. Few steps on extraction of PHA compound that involves chemical, physical, mechanical, and biological method. For discussion, graph showing the growth kinetics of algae between the cultured broth and non-mixed broth. Using the spectrophotometer, optical density was calculated. The growth curve of Chlorella pyrenoidosa with the presence of organic compounds which are nitrogen, phosphorus, and potassium keeps exponentially increased after day 6. Meanwhile, cultured broth, the absence of organic compound started to slowly reach stationary phase. This indicates that the cultivations of microalgae needed a boost on the concentrations of organic compound, yet still able to adapt even with limited amount of nutrient. Next, graph showing the effect of pH and temperature on the productions of PHP during the cultivations of microalgae. Lower acidic pH resulted in significant decrease in production compared to higher alkaline pH, where it remains stable. From the graph, the maximum or optimal productivity can be seen at 40 degrees Celsius at pH 7.5, where the productions of PHP showing the maximum value. This indicates that even with the fluctuations of pH and temperature, microalgae is still able to produce desirable amount of PHP. In conclusion, the laboratory testing on the growth rate of cultivation microalgae can be facilitated. When optimal requirement is achieved, microalgae can produce a significant amount of PHA. This supports that microalgae can be used in biomass production of bioplastic. Continuous research and study need to be done to lower down the number of plastic in the environment. These are few references that I have included for my article review. And that's all from me. I will genuinely accept if the panels have any question regarding to my presentation.
Okay, thank you, Akmal. Any question from the panel?
Okay, Akmal, since nobody asked, what do you think about the durability of this algae to to replace the plastic?
To replace, sorry?
You are going to find an alternative to replace plastic, yeah? Yeah?
Mm-hmm.
Okay, what do you think about the durability? Ketahanan.
Oh, okay. The durability, I think the bioplastic, the chemical compounds of the bioplastic and plastic is slightly similar with the plastic that we use in nowadays. The difference is that the bioplastic is really good for biodegradable, really good in properties of biodegradable, so that it can be degrade in the soils. That's all for me.
Okay, thank you.
Okay.
Okay, Rahmal. It is continuity question from Dr. Siti Khairia. A question, because the question is about the durability and also the strength actually for your materials from bioplastic, because if you compare bioplastic and conventional plastic from the biofuel or conventional plastic uh, from fossil fuel, I think. Okay, it's not comparable actually, because from bioplastic the mechanical strength is very low and it will limit the application. So what your opinion on this?
My opinion is that we need to continue our research and studies on this biodegradable plastic, so that we can improve the structure for the plastic to be used in the future.
Okay.
Okay.
Okay, that's all from the panel. Thank you, Muhammad Nur Akmal.
Masa ada lagi tau?
I think dah lebih 2 minit. Untuk Q&A ke?
No, sebab Akmal habis cepat tadi.
Oh, sorry.
Belum 8 minit pun lagi.
Oh, belum 8 minit lagi.
Yes.
Saya tak dengar bunyi. So,
Satu lagi Akmal, I think you are not allowed to use the logo, the Jata logo.
Oh.
Because it's not official, the Jata logo. Your item logo is sufficient enough.
Oh, okay. Sorry for that.
And is it the only references that you have for this review?
Actually, there in my article review, there is a lot of references, but this is, as I stated before, these are some of the references from my article research.
I think there is no problem. Just list all your references, because we want to know how many references that you use for this review article. Because for review article, normally we need lots of reference.
okay, itu saja ya. Hopefully nanti Akmal boleh improvise, but I think you have the full number of reference in your article review submission later, yeah? Only for this presentation purpose. Okay. Thank you, Muhammad Nur Akmal. Okay, can we have the next presenter, Nur Akashah Amran?
Yes, I'm here.
Okay.
Can my slide be seen, Doctor?
Okay, clear?
Okay. Assalamualaikum and a very good morning to all my panel lecturers and my fellow friends. So my name is Nurakasha binti Amran and I'm going to present my final year project with the title of Fruit Preference and Feeding Behavior of Aldabra and Elongated Tortoise. So let's jump on the quick review. This will be the contents of my presentation for today. So without further ado, I will move on to the introduction with my background study. So my study subject in this research are tortoise from reptile species. Both species involved, which is Aldabra and Elongated, are endangered species. So in terms of food, tortoise in wild habitat, majority are vegetarians. They feed on fruits, flowers, leaves, and vegetables. During the year, some species are known to eat up to 200 varieties of plants. And then moving on to the problem statement, we know that there are two types of animals. One in the wild, in the habitat, in their own habitat. Another one was bring to be kept in captivity, like in farm or in zoos. So when tortoises are kept in captivity, there will be issues that will arise from time to time regarding food and their diet. Some of the problems are like individuals of tortoise not getting enough the same amount of food, competency between tortoise, and also the dominance factor. Captive animals are normally fed depending on the farm or zoo knowledge, but food products and composition differ greatly between zoos and do not comply with any recognized standards. This means that not all zoos offer the same diet and the same nutritional value for the tortoise. Tortoise in captivity might have their own preferences, but since they live in captivity, they have to survive with the food that were offered to them. So the significance of my study is by studying the food preference of tortoise, zoos can provide and prepare better diet for the tortoise according to their preference, as well as they can avoid food wasting when they know that tortoise didn't prefer some type of food. And then the second one is the observation of feeding behavior can make zoo opted for other options in placing the tortoise in captivity by taking into consideration their behavior and interaction during feeding so that all tortoise will get enough amount of food for their growth and survival. These are the two objectives of my study, which is to determine the most preferred food for Aldabra and elongated tortoise, and the second one is to quantify the feeding behavior for Aldabra and elongated tortoise. So moving on to my methodology, I did the study in KLTMZ, which is the Kuala Lumpur Tower Mini Zoo, and then the subject for my research is two species, which is Aldabra and elongated, with 3 and 9 individuals respectively were used in this study, which means there are 3 Aldabra tortoise and 9 elongated tortoise. The Aldabra tortoise was housed in 22 times 4 meter square cage. The 9 elongated tortoise were housed in another cage, which is 13.5 times 2 meter square. This is the grouping for my research. I did 4 groups. I divided the tortoise into 4 groups, which is the first one is the female Aldabra, which is the average of 88.3 centimeter measurement. I did the measurement from head to tail. And the second one started where elongated tortoise there from group 2, 3, 4, the 9 elongated tortoise is placed, which group 2 with average of 37 cm, group 3 with average of 22 cm, and group 4 is average of 18.6 cm. And then the experiment timing is done in 30 minutes. For all Aldabra tortoise, we start at 9 a.m. and elongated tortoise start at 4 p.m. This is the flow chart of my methodology. First, I prepared food in separate trays and weighed them first. And then I set up the camera. And then I made sure the tortoise is in their own housing system. Then I recorded the time spent consuming on each food. And then feeding behavior was recorded on live observation. And then after 30 minutes, the final weight of the food is weighted, data is collected, and then I did the statistical analysis. The methods for behavior for the objective 2, which I need to see the feeding behavior. There are 3 behaviors that I observe, which is pinching, retracting, and aggressive interaction. Pinching is when they are fully raised head and body while stationary, and then retracting is when they pull their head back into their carapace, and then aggressive interaction is when they are being aggressive to another tortoise, which is they are biting another tortoise. This is the example of their behavior: pinching, aggressive, and retract. And then all group were observed for 10 days in total. And then I analyzed the data in using Wilcoxon test, ANOVA, and Poisson regression. moving on to the result and discussion. The first one is for my first objective, which is to determine the most preferred food for Aldabra and elongated tortoise. So this is the graph. As you can see, I used the Wilkerson sign writers, showed that the feeding time of tomato and carrot elicit a statistically significant difference in individuals because p-value is 0, which is lower than 0.05. For time spent on consuming food and weight of food consumed, normality test was conducted for both data. And with the p-value of 0, the data are not normally distributed, therefore Wilkerson test was used. In general, regardless of the group, the tortoise significantly spend more time feeding on tomatoes compared to carrot. As seen in the mean between those two food, time spent feeding on tomatoes was relatively higher than carrots. Next graph is also for the first objective, which is the weight of food consumed. This I did the test of normality, and it is shown to be significant. The first group is significantly different from any other group. As seen in the table on very below right of the slide, according to Wilkerson sign rank test, we can see that group 1 is the only different one from the other group, because group 1 consists of 3 Aldabra tortoise, which is way bigger than elongated tortoise. So they eat more tomatoes and more carrots compared to the other groups. So for my discussion on the first objective, since the items and food tend to be hard, initiating a crack is usually a problem. Fracturing such food can require constant work. Aldabra giant tortoise eat primarily grass, leaves, woody plants, so they may have experiences in swallowing and consuming hard food, which is in my study is carrot compared to tomatoes, which is more mushy and softer. So the second objective is to quantify the feeding behavior of Aldabra and elongated. So the data I present here is the feeding all, which is the time spent feeding regardless of time of food, and I use the ANOVA test. P-value is 0. Data is statistically significant. Post hoc test showed different p-value and mean value between group. Overall, from post hoc test, group 1, which is Aldabra tortoise, spends significantly higher time on feeding compared to elongated tortoise in other groups. This is a discussion on why group 1 has spent more time compared to other groups. And then this is the feeding behavior, which is the objective 2, to see the feeding behavior of the Aldabra and elongated tortoise. I did 3 behaviors and I observed 3 of them, which is retract only shows the significant data. So for retract, also this one is group 1 showed significantly different data from other groups because they recorded the highest frequency of retracting behavior. The label as A, B, and B for the graph is the group 1 is different from group 3 and 4, as well as but the group 2 is standalone, and therefore they have been labeled as A and B. So tortoises retract their head into the carapace while they feel threatened, and when they just try, turtles use their flexible neck to retract their head safely inside their shells. So as a conclusion, elongated showed preference to tomato compared to carrot. However, Aldabra showed no preference as they consume both food and with the same amount. They also spend more time consuming tomato and carrot. Overall, group 1 spent significantly higher time on feeding compared to elongated tortoise and other group. Aldabra tortoise also recorded the highest frequency of retracting behavior. And my recommendation is, as I mentioned before, the zoo has to opt for other options for placing the tortoise in captivity by taking into consideration their behavior and interaction during feeding. Zoo also opted for another type of vegetable that has the same nutritional value as carrots, since the observation showed that the tortoise do not prefer carrot as much as they do on tomato. And then this is the list of my references. There are 20 references in total. That's all from me. Thank you. I welcome any questions.
Okay, thank you, Nur Akasha. Any question from the panel?
Akasha, I have like a comment. Probability value never equals to 0. If you find it's 0, it means that it is too small. It is maybe less than 0.001 or less than 0.00001. So never put equals to. Okay?
Okay, Doctor. I will correct it in my final report.
So Akasha, do you think that the behavior that you study during your final year project has to do with the food preferences for that Aldabra and elongated tortoise?
I think they are somehow related because they are placed in the same position, in the same cage, so they are, because I also observe the aggressive interaction. So sometimes when one of the tortoise has finished their food, they tend to move to the other tray for that I prepared for other tortoise. So there will be aggressive interaction where they will be biting because they are trying to eat the food from the that I prepared for the other tortoise.
So because you are studying the two different species, right?
Yes.
Same two different species. So do you think that the behavior also is contributed by by that different species? So it's like
Oh no, because
inter-specific competition for that food, or it
uh,
just that.
I think they are related because when they are placed in the same place, as I said before, two different groups will interact with their own species. Because as I said before, I placed three elongated tortoise in one group, three Aldabra tortoise in one group. So they interact and have interaction between their own group mates.
Okay. Mm, anything from Dr. Siti Nur Asma?
Okay, Akasha.
Yes.
I just want to comment on the graph because most of your graph there is no units. You can refer back to your graph, and also make sure the decimal numbers also in the same decimal
Standardized.
number. Yeah, standardized. Okay.
Okay, doctor. I will take note on that. Thank you.
Okay. And this research also did by you, right? This is
Yeah, this is
not a review research. Okay, because in the discussion, the first objective and the second objective, you mentioned some discussion on the objective, but you quote another person work, okay, other previous researcher. So in the discussion, please discuss your finding, okay, because this research done by you, so you have some result to discuss.
okay, doctor.
Okay.
Okay, thank you, Akasha.
Thank you, doctor.
Next we have Afimi Effendi.
Can you hear me?
Okay,
All right.
clear.
Okay, Assalamualaikum and a very good afternoon to all examiners and my fellow friends. I'm Afimi Effendi Malik. I complete my final year project under the supervision of Dr. Faizal Pardi, and which entitled Community Structure and Biomass Distribution of Three Species at Compartment 85 Sungai Lalang, Forest Reserve Selangor. So let's get started. So Sungai Lalang is one of the tropical rainforests at Peninsular Malaysia, and one of the problem is the recreational activities. So Sungai Lalang is well known for its rivers, namely Sungai Gabai and Sungai Tekala, which attract visitors especially on weekends and the holiday seasons. Activities such as jungle trekking, camping, picnicking does negatively affect Sungai Lalang forest ecosystem. According to Duffield and Walker, recreational activities cause detrimental effect on vegetation, disturbance to wildlife, soil erosion, as well as water pollution and increased fire frequency. So this study is important to provide forest inventory and significantly beneficial in contributing to sustainable development goals under the SDG 15 Life on Land. Um, okay, we move to the objective. I have three objectives of this study, which is first to quantify the tree species composition at Compartment 85, Sungai Lalang. Secondly, to determine the species diversity, and thirdly, to estimate the biomass distribution of tree species at Compartment 85, Sungai Lalang, Forest Reserve, Selangor. So, methodology. Due to COVID pandemic in Malaysia, the sampling activities could not be implemented because interstate travelling is not permitted. Therefore, secondary data of forestry composition in Sungai Lalang Forest Reserve was obtained from the Forestry Department of Peninsular Malaysia. So this is my study site, which is located at Sembilan, Selangor. And then we move on to the methodology. So basically, this is the method used by the foresters to identify the community structure and biomass distribution of the study. So first, a quadrant was established with an area of 1 hectare, and then the DBH, the meter breast height of 5 centimeter and above were enumerated or were measured, and then the sample was collected, and then the tree species was identified, and this is where I come to do the work, which is doing the data analysis, which I divide or classify into 3 components, which is taxonomy, composition, tree diversity, and forest structure. And then we move on to result and discussion. First, on the taxonomic composition. After the analysis, I conclude that the taxonomic composition of this forest was recorded by a total of 356 trees from 32 family, 51 genus, and 51 species. So Leguminosae was recorded as the most specious family with 7 species recorded, and Citrus SPP was recorded as the highest individuals or species with the most individuals with 58 individuals. So in this table, you can see that I compare it to another forest site, and as you can see, this forest Sungai Lalang, this study do not have taxonomic composition as high as the other forest sites, because during the analysis, the species most of the tree species were not identified. Most of the trees were not identified up to species level. And then we move on to the species richness, diversity, and evenness. So, species Shannon index, diversity index usually lies between 1.5 and 3.5 according to Maguran 1988. And evenness value ranges from 0 to 1, 1 being the highest, and it means that all species are equally abundant. So from this study, it is recorded that the Shannon index for this forest is 3.13, which is generally high, but in comparison to the other forests, such as Bukit Pancho State Park and Borobelum State Park and Pulau Jarajak Forest, it is kind of low due to the species, most of trees that is not identified up to species levels, which is, I can say, one of the limitation of this study. And then we move on to the forest structure. To understand the forest structure, I divided the tree diameter at breast height into 8 classes. The stand structure of the study consisted of 356 individuals and was illustrated by the inverse J-shaped pattern. However, there is an anomaly in its strength as 77 individuals dominated the DBH class 5, ranging from 45 to 54.9 centimeter. So the graph also shows that Sungai Lalang was dominated by saplings and small trees, which shows that Sungai Lalang is an actively regenerating forest based on the sustainable forest management practices. So next we'll move on to the family importance value index. So importance value index, this is the rule. So family with more than 40% and species with more than 10% can be considered as having absolute dominance. So we can see that in the in my study area, there are no family that is having absolute dominance towards the study area, because the importance value index does not exceed 40%. So, but it can be said that all three families listed were relatively dominant in comparison to the other families. And meanwhile, for species, Elateriospermum tapos was recorded as 30.3% importance value index, which is So we can consider it as species with importance value index. As having absolute dominance toward the forest because the value exceeds 10%. And then we move on to biomass estimation, which is 50% of the weight of tree is made up by carbon, according to Basuki. And the total from the analysis, the total biomass of 1 hectare compartment of Sungai Lalang was estimated at 480.6 ton per hectare, which was contributed by the above ground biomass and below ground biomass at 403.1 ton per hectare and 67.5 ton per hectare, respectively. And then the high biomass portrays by the rapid growth and regeneration of living tree biomass and carbon accumulation within the study area. And then, in conclusion, all objectives that I proposed in the study were met. All species were able to be quantified. The species diversity were able to be determined, and the biomass distribution were estimated at 480.6 ton per hectare. So there are a lot of limitations in this study. So I propose a few recommendations, which is first, the trees should be identified up to species level during the identification process to obtain more accurate and reliable results. And then the forester can include other parameters such as adaptive factor. And lastly, I recommended that the forest management team to closely monitor the condition of the forest based on the study to implement forest restoration. This is my reference list. And that's all from me. Thank you. I welcome any questions.
Okay, Assalamualaikum Afimi. Boleh tanya dah Kak Izzah? Okay.
Boleh.
Okay, Afimi.
Yes.
I just want to ask regarding your data. You kata you couldn't identify to species level, so you only managed to identify 51 species.
Yes.
So, but you collected all data. I mean, you collected all leaves ke apa, right?
This data is a secondary data, which is
secondary data.
Yeah.
Sorry, Dapat Izzah lupa lagi. Okay, so from that secondary data, the person collecting the data only managed to get out that 51 data.
Yes.
Okay, never mind. Okay, thank you. Sorry. Okay.
Afimi,
Yes.
why do you think that the diversity at the study site is low as compared to the other forests?
Oh, because first I think it's because of the limitation which the species were not identified to species, all the trees were not identified to species level. And I think secondly, if we compare to the other forests that I compared, such as Bukit Pancu State Park, but Bukit Pancu State Park has a higher diversity because it is a state park. So the area is protected by the state government. So which makes the forest able to maintain its high diversity. The next one is Royal Belum State Park, which I compared. So Royal Belum is a primary forest or a virgin forest. So it has less or no disturbance from the anthropogenic activities or natural disasters. And the third one I compared to Pulau Jerejak. So Pulau Jerejak is away from the mainland. So it is an island forest ecosystem, which makes it has less disturbance and less development surrounding the forest, which contribute to high diversity.
Thank you. Very good answer there.
Is there more time?
No.
Okay, bye.
Okay. Thank you, Fimi. Okay, next. Can we have Siti Nur Alia Anis?
Yes, doctor. Can you see my slide?
Still loading. Okay.
Okay, Assalamualaikum and a very good evening to respected lecturers and my beloved friends. So my name is Siti Nuralanin Salahuddin and I would like to present my final year project which is entitled Quantifying Forest Structure and Species Richness of Tree Communities at Compartment 27, Sungai Lalang Forest Reserve, Selangor. So this is the overview of my report. So we move on to the first chapter. Okay, the background of study starts with the Asian tropical forest has faced much more modification compared to the one in Africa and South America. This has led to the statement by Genevet 2020 that forests are progressively fragmented by human activities such as overexploitation or harvesting beyond sustained yields and forest land conversion to forest products such as lumber, paper and furniture. Also, this study takes place in precious tracts of remaining tropical forest in Selangor, which is Sungai Lalang Forest Reserve. So, I would like to state that problem that I met in this study. I have found the forest statistic that they show decline in forest area from 40.16 million kilometers squared in 2010 to 39.96 million kilometers squared in 2016. Also, we can see that the decline in
Sorry, Alia, your slide is not moving.
Oh, okay.
Okay.
I have shared the window, not.
Yes, you should share screen.
Entire screen. Okay, I'm sorry. Can I start back?
Okay.
Okay. Can you see my slide?
Can you do the slideshow mode? Okay.
Okay, it's moving, right?
Okay.
It's moving.
You can just continue from the last slide.
Okay. For the second problem statement, which is there are decline in area of Sungai Lalang. This because this area can be accessed by many types of vehicle and surrounded by housing estates, oil pump plantation, factories, highways, and the new township. So the objective for this study is to quantify tree species composition and species diversity at Compartment 27 at Sungai Lalang Forest Reserve. The second one is to estimate the total biomass of tree communities at Compartment 27 of Sungai Lalang Forest Reserve. So we move on to the research methodology. So due to the pandemic situation, we having the limitation for the sampling activities. So the secondary data of floristic composition in Sungai Lalang Forest Reserve was provided by Forestry Department of Peninsular Malaysia. So the research start with the quadrat sampling, which is 10 plots of 0.1 hectare each with 50 meter times 20 meter, and then ended with the data analysis of taxonomy composition, tree diversity and forest structure data including biomass were analyzed. So the method number 1 until method number 4 was conducted by Forestry Department of Peninsular Malaysia itself. Only the method number 5 is conducted by me. Then the result discussion, we start with the taxonomy composition. Sungai Lalang Forest Reserve has enumerated 48 tree species in 44 genera and 20 families. We can see the comparison between all the different forests in Southeast Asia. We can see that Sungai Lalang Forest Reserve has obtained lower taxonomic composition. This is due to this forest has been changed from virgin forest into secondary forest due to numerous development activities. Then we move on to the species richness, diversity and evenness. Okay, we can see that this forest has obtained 3.09 and it considered high and within acceptable range. But we obtained the low evenness because I believe that some species is not similar in number. Okay, then we move on to the DBH, the meter at breast height distribution. We have obtained the inverse J-shaped pattern with the denomination of 15.0 cm until 25 4.9 cm. And this inverse J pattern is in line with the other previous study. Then we move on to the abundance parameter. This is the brief information for abundance parameter. So in terms of frequency, top leading species was Cinnamomum iners, which has a total of 100% frequency. So this is the picture of Cinnamomum iners. So in terms of basal area, we can see that Diptera has the highest basal area. This is because most of the trees in the family Diptera has the largest size and largest DBH. Then we move on to the important value index. We can see that also Shoria sp2, which is from Diptera also show the highest importance value index, and it has achieved absolute dominance in terms of species because it exceeds the 10% of species importance value index. Okay, for the biomass estimation, the total biomass estimation for Sungai Lalang Forest Reserve is 651.55 trees per hectare, with above ground biomass of 557.38 trees per hectare, and the remaining which is below ground biomass of 94.17 trees per hectare. So we can see that this from the above ground biomass, it contributed by stem, branches, and leaves. So this picture is show the Shoria parvifolia trunk, which is contributed to the above ground biomass. Also, Shoria sp2 show the highest biomass in terms of species. This is because Shoria sp2 has the largest DBH in this study, which is 106.9 cm. So to conclude, the two conclusion has met the two objectives. So the first objective, which is to find the tree species composition and species diversity. So I have enumerated that this forest has 48 species and 44 genera from 23 families, and the value of Margalef is 8.09, Shannon Winner 3.09, and low evenness value of 0.46. Then the second objective, which is to estimate the total biomass. So the conclusion for the second conclusion is the total biomass in this forest is 651.55 trees per hectare. So for this study, the recommendation is first that we must identify all three species at species level in order to obtain more accurate result. Then I would like to recommend that soil property study can be done in the future, because there are many studies that show the positive relationship between soil properties and species composition. The last recommendation, I would say that the importance of conservation of Sungai Lalang Forest Reserve must be highlighted because it has the potential for seed production, gene bank, and tree conservation. So this is my references, and that's all for me. Thank you. I would like to welcome any question.
Okay, Siti Alia, thank you for your presentation.
Okay.
Not much to ask, but can you just go, alamak, you dah tutup ke presentation? Can you just go back to your results, the diversity part?
Right, I will share it again. Diversity, okay.
Result. Yang sebelum ni, okay.
Sorry.
Aku tak nampak. Sini kot lah, sini. Okay, you ada mention tadi, you have mentioned tadi, but just wondering again, can you explain agaknya lagi sekali, why is the what may contribute to the low diversity of the family apa semua in your study area?
First, because of the limitation of the sampling activity.
Uh-huh.
So the criteria that all the species is not really identified to species level.
Not the species level, I tanya about family. Why do you think the family also is less in that area? I mean, I understand the species level problem, but why is it agaknya there are only like 23 families were found?
I think because the status of Sungai Forest Reserve has changed from virgin forest to agriculture and residential area to the numerous development activities such as Sungai Tekala, recreational park. So it may contribute to the low family distribution in that forest.
So do you mean that if there's due to the human activities, so they only cut off a few family trees?
No, I think in overall, maybe the family who are really adapted, I mean like really robust and can adapt to the changes in nature, they can survive more.
So is it not possible that maybe perhaps because of some other environmental factors like soil or that kind of thing, that only a few species that is, only a few species that kind of like adapted to that area?
Yes. I think there's also a possibility because different species has different tolerance to some environmental factor.
Okay, thank you.
Thank you for the question. Thank you.
Okay, any other question? But we are actually exceed the 2 minutes session of Q&A. Tak ada ya? Okay.
I have no question, but I like your slide design.
Oh, thank you, Doctor. Thank you, Prof.
Okay, thank you, thank you, Alia, Siti Nur Alia.
Alright.
Next, can we invite Cik Nur Izzati? Okay.
Yes, I'm here.
Dr. Faiza, is it okay for me to just pesan jangan tutup slide dulu in case kita nak tanya based on the slide, the presentation, okay?
Okay, boleh?
Can my slide be seen?
Yes.
Can I start now? A very good evening, Abi, to all the panels and all my fellow friends here. My name is Cik Nur Izzati binti Muhammad Najib, and today I would like to present my thesis entitled Diversity of Order Diptera in Selected Forests at Pulau Tuba. First and foremost, for some introduction, according to Baron et al., insects from the class Insecta lead the terrestrial ecosystem in terms of species richness among all arthropods in the global biomass distribution with about 1 million species. And Diptera is one of the 28 orders that are classified under class Insecta, which the word Diptera comes from the Greek words di, which mean two, and ptera that refers to the wings, meaning that only a single pair of wings are functional in the adults of true flies. Next, this study was conducted in Tuba Island. Tuba Island is an island that is very rich in flora and fauna, located 5 kilometers southwest of Kuajiti, which is the main terminal ferry in Langkawi. And the Tuba names, the name of Tuba comes from the root of Tuba itself that can be widely found on the island. The objective of this study is to quantify the diversity of order Diptera at selected forests of Pulau Tuba, as well as to study the relationship between order Diptera's diversity with the variation in the environmental gradient from the forest fringe to the inner forest. First and foremost, Diptera is known to have a lot of roles and functions in the ecosystem. Diptera firstly act as a decomposers where they produce the nutrient-rich soil layer that helps in growing plant. Not only that, the larvae stage also contribute to organic materials decomposition as well as supply nutrient for plants. Next, aside from bees, moths, and butterfly as pollinator of flowers, most people do not know that flies are second in importance to bees as a pollinator. Not only that, Diptera also act as parasitoids, where Tachinidae, Bombylidae, Phoridae, Sarcophagidae, and Callipoidae are the families of Diptera that often be mentioned as a parasitoids. And Verkroth also said that the flies are the most effective and important species for forensic research, where the forensic researchers will predict the post-mortem index or any alteration in the location of the body as well as the cause of death by just looking at the species of Diptera available at the location. For the methodology of this study, first and foremost, the sampling activity was conducted by Dr. Siti Khairia binti Muhammad from 17 September to 21st September in Tuba Island. For the method, firstly, the Malay trap were set up, where the arrangement of the traps at each transect is kept at 100 meters between each other, starting from the forest fringe to the inner forest. Next, for insect sorting, it is done in a laboratory of UiTM Shah Alam for 6 weeks using the stereoscopic microscope. As for the mounting and preservation step, the specimens of insect were pinned using an insect pins to the mounting board, and it is dried in oven to prevent from fungal. After that, family identification were conducted using a reference book, and data analysis were done using PASS software, where we want to find out the Shannon-Warney Diversity Index, Evenness Index, Margalit Index, and also by using SPSS, Kruskal-Wallis H-test, and pairwise comparison were done. As for the overall results, a total of 5,450 specimens were collected, where 18 families were identified together with 27 morpho species recorded. As you can see, figure 1 shows the percentage of each Diptera family found at the study site. Mycetophilidae is the highest family found at the site, followed by second in line Scaridae, and then Poridae recorded as the third largest family found. This result is supported by Jeskov and Hipa, saying that genus Manuta, which is under the family of Mycetophilidae, are one of the main forest residents, even though the adults are quite inconspicuous to their habitats. And also, a study from Kitching support this result, saying that Scaridae and Poridae were the common largest family found in the study conducted in the forest. While Anisoporidae and Drosophilidae rank as the least family recorded from this research, with the percentage of 0.20% and 0.18% respectively. This might due to Anisoporidae is only a small group of gnats from the family of Diptera, with only about 100 species worldwide found. As for Drosophilidae, they are known to be the pest in household and commonly found in household with the presence of fruit. The larvae of the Drosophilidae feed on the yeast growing in the fruit. As you can see, table 1 shows the distribution of Diptera across 3 sampling plots. From the table, the highest morpho species recorded are from family Scaridae and family Tabanidae with 3 morpho species each, while the highest individual is Mycetophilidae with 2,736 individuals, and the lowest individuals is Drosophilidae with only 10 individuals recorded. Trap 1 with the greatest number of insects among all traps now is located at the forest fringe, and as we know, forest fringe had the great presence of flowering vegetation along the forest fringe itself. So because of that, Diptera were drawn to there because of the shrubs and flowering vegetation that serve as a food supply for them. Also, the habitat preference of Diptera towards the humidity, where forest fringe had great availability of sunlight. For Trap 3, the smaller number of Diptera in Trap 3 were expected due to inner forest is densely forested. As we know, inner forest consists less shrub, less weeds, and less flowering vegetation that can be consumed by the family of Diptera. So also due to the less number of Diptera collected, also due to certain species are not numerous during the sampling time, which is only for 5 days. As you can see from this slide, table 2 show the diversity indices of each study site. From the table, Trap 1 show the highest Shannon-Warney Diversity Index with H equals to 2.05, and also the highest Margalit Index with R equals to 1.91, even though the Evenness Index here is moderate compared to the other trap. This shows that Trap 1 have the highest diversity of insects and rich in Diptera species, even though the distribution of individuals among different families are not even. While Trap 3 had the highest Evenness Index recorded, but moderate in H and R value. This indicates that the distribution of individuals of different family have similar distribution, even with low individuals and species. This is the result from the Kruskal-Wallis H-test. From the result, we found out that the distribution of the family of Diptera across all traps was significant, and the family's distribution differ between each location. The difference comes from Trap 1 and Trap 2 with P equals to 0.028, also from Trap 1 and Trap 3 with P equals to 0.033, and also from Trap 2 and Trap 3 with P is lower than 0.01. As the conclusion, we found out the forest fringe is highly abundant and diverse in Diptera species compared to the middle and inner of the forest. The result from this study did not support the hypothesis of the equal distribution of Diptera between tree traps when the P is lower than 0.05. As the recommendation, in 10 years' time, I hope that similar study can be made in this location to find out whether global warming and climate change will affect the diversity and abundance of Diptera in this location, as well as more study on Diptera diversity can be made with greater duration of study after the COVID era so that every species collected can be identified properly. These are my reference for this slide. And with that, I end my presentation. Thank you.
Okay, thank you, Cik Nuriza Izzati. Can I ask you why you mention your title as selected forest in Pulau Tuba? Because I thought Pulau Tuba supposed to have like its own one entity forest, namely Pulau Tuba Forest Reserve.
Okay. For the question, even though the sampling, this sampling activity were done by Dr. Siti Syariah, but I think the reason why I put the selected forest because we don't count the whole forest of Pulau Tuba. We only take from the forest fringe to the inner forest in the location of 100 meter between each transect of Malay strap. So it might not cover the whole result for the forest in Pulau Tuba.
Okay, I think all the ecologists, maybe the entomologists, we can never sample like the whole area of any forest or any places, right? Because it will take like maybe more than 1 year to compile all the results. So I think you can just put your title as apa tadi? At Pulau Tuba Forest Reserve, because I know Pulau Tuba has its own macam satu signage tu, the name of that area is Pulau Tuba Forest Reserve, because the way you said selected tu dia macam ada, if you said like selected forest in Langkawi, yes, correct, because there are many forest reserve, tapi if it's only in Pulau Tuba then memang Pulau Tuba satu lah, but we understand you cannot sample every single part of Pulau Tuba.
Alright,
Okay. And number two, how if you combine data result from all traps? Because just now you just present your data individually from trap 1, trap 2 and trap 3 kan? Trap number 3. So I would like to know how if you combine all of them. Perhaps you can get like more more diversity of that insect.
Because the objective of my study is to find out the diversity of insect at different elevation of forest.
Oh, okay. So different trap was located at different elevation, eh?
Yes.
Okay. So,
Can I ask one more question? Sorry,
boleh,
Kak Faizah, nak tanya lagi?
Boleh. Sila.
Okay, so Izzah, just wondering, because you mentioned on the comparison between trap 1 and trap 3, with trap 1 is that the tree inner forest, so less food to consume, blah, blah, blah. But as we can see from the crustal wallis, that the trap 2 yang sebenarnya very different from trap 1 and trap 3. So why do you think it's different? Why do you think the middle of the forest is different from trap 1 and trap 3?
Okay, for this question, I think that might due to the between the traps, between the traps is only 100 meter, and that might be too short for in us to calculate the diversity, because might be that the middle forest might be the forest fringe that is might be the insects already went to the forest fringe, because the it is very short, the location between transect 1 and transect 2.
Ya ke? Okay, tak apa, terima. Okay. Okay, thank you. I want to ask lagi tapi macam tak dapat kan. Khairia, habis masa ya?
Dah lama habis.
Okay, bye.
Okay, thank you Cik Nur Izzati.
Alright.
Okay, can we have Wafika Ismail?
Yes, saya.
Okay.
Okay. I'm ready to start now. Assalamualaikum and a good afternoon to the panel of examiners, my supervisor and my final year project colleagues. My name is Wafika binti Ismail and I would like to present on my final year project with the title Diversity and Distribution of Hymenoptera in Selected Forests of Tuba Island. First of all, a basic idea of insects, Hymenoptera, and the study site will be explained. Insects are a highly successful animal that act as an essential agent in the agricultural and ecological system. This is for the life cycle of living things or as parasite in crops or domestic stocks. Under the class Insecta, order Hymenoptera is observed by entomologists due to its abundance, which this order comprises about 20% of the insects worldwide. For the study site, Tuba Island is 9.26 km from Marble Jetty Kuah with a total area of 504 hectare accumulating 62.78% of Langkawi Archipelago. The objectives of the study are to quantify the abundance and diversity of Hymenoptera in selected forests of Tuba Island. Next, it is to differentiate the species composition of Hymenoptera from the forest fringe to the inner forest. Then the literature review will be focused on ecological and economical impacts. The ecological impacts of Hymenoptera as a decomposer and contribute to the food web's process in the ecosystem. We do this by decomposing the tissues of an organism and cleaning up the forest litter. As a pollinator, bee is essential to sustain a healthy ecosystem. This is because many flowering plants rely on bees for pollination. In turn, it will provide food and habitat for a range of other creatures. For the economical impacts, the production of honey and honey byproducts from honeybees for the source of income and food has shown increase the profitability for bee farmers and the creation of traditional medicine in rural area as stated by Bata et al. In 2014. Next, as parasitoids, they are used as a biocontrol agent to save crops from other insect invaders and reduce the usage of insecticide. The sampling activities were conducted by Dr. Siti Haririah from 70th to the 21st September 2020 in Tuba Island. As seen in figure 1, the Malay strap were positioned at 100 meter from forest fringe to the mid-forest and 100 meter from the mid-forest to the inner forest accordingly. Moving on to the methodology in figure 2, the first step was setting up the Malay strap. Then the sample were collected from killing bottle which were filled with 70% alcohol. The samples were then mounted and preserved in biological laboratory. The family identification was using a reference book which is Hymenoptera of the World and Identification Guide to Families. The data analysis was then performed using Shannon-Wiener Diversity Index, Evenness Index, Margolef Index, and Kruskal-Wallis test using SPSS version 26. The overall results obtained showed that a total of 150 individuals gathered, with a total of 32 morphospecies recorded and a total of 12 families identified. Figure 3 showed the percentage and the family's name identified. Based on results and discussion, the families of Formicidae, Braconidae, and Ichneumonidae had the highest number of individuals. Formicidae was the most abundant with 29% as they live in colony and essential as predator, decomposer, herbivores, and bioindicator for the health of agricultural ecosystem. Braconidae with 19% and Ichneumonidae with 18% were parasitoid Hymenoptera family as they have the highest number of species and they dominate the forest ecosystem. While the least abundant families were Megacelidae, Pteromalidae, Tenthridinidae, and Vespidae. This indicates that the study site was not suited for the species habitat due to anthropogenic disturbance or temporal differences during the sampling related to rainy seasons. Based on table 1, track 1 was denoted as forest fringe, track 2 as mid-forest, and track 3 as inner forest. Track 1 accumulated the highest percentage of individuals with 43.71%, and the percentage decreases as track increases. Based on discussion, similar findings were found in families of Hymenoptera in Langkawi, conducted by Siti Haria et al. In 2014. This is because they have homogeneous climate and Tuba Island is in Langkawi archipelago. Track 1 recorded the highest number of individuals as the presence of flowering plants near the forest fringe serve as food sources for Hymenoptera family. Track 3 recorded the least number of individuals as fewer flowering plants were accessible and the inner forest was densely forested. Further data analysis of diversity indices is shown in Table 2. Track 1 recorded the highest H prime value with 1.91 due to the abundance of flowering plants contributing to the diversity of species. The E prime value was highest at track 2 with 0.83 as it was evenly distributed but has lower diversity. The R prime value recorded the highest value at track 1 with 2.86, specifying a high species richness compared to the other two traps. As a conclusion, Hymenoptera were most abundant near the forest fringe of Tuba Island, indicating a high diversity, and the diversity of Hymenoptera differs significantly between the forest fringe, mid-forest, and inner forest, where the significance value is less than 0.05. And as a recommendation, further studies are required by increasing the duration of sampling and increasing the number of Malay strap in order to gather a higher number of samples in Tuba Island. As well as sampling can be conducted based on temporal differences, such as according to types of monsoon in Malaysia. And these are all the list of references that I have used in this slide. Thank you.
Okay, thank you, Wafika. Again, same as the previous presenter, I would like to advise you to just put the title of Forest of Pulau Tuba instead of selected forest, or perhaps you can put macam at Island Forest of Pulau Tuba. Dia nampak lebih promising because you're study in island, which is coastal forest, and coastal forest should have macam unique diversity index compared to inland forest tau. So maybe in your discussion nanti in thesis you can describe why you have like low H prime, low ada evenness ke? Tak ingat tadi. H prime ke, low evenness at mana fringe, inner and outer, because in island ecosystem they have macam they have their own distance kan from the mainland. Kalau macam you have extra time to make a comparison with other inland forest pun, you can see that the inland forest compose more species compared to the coast island forest ataupun coastal forest. Because island ni dia interesting tau in terms of the presentation of species composition, tree species and also the fauna lah.
Noted.
Okay. Any other panel? Any other question from other panel? Tak ada ke Dr. Siti Nur Asmah, ada tak?
Okay, Wafika. May I know what is the major contribution of your study to this, maybe to this insect family or the forestry?
Sorry, can you elaborate the question?
Sorry?
Can you elaborate the question in terms of?
Okay, yeah. What is your major contribution of this study? Okay, to the forestry or to the insect family that you studied before?
Based on my research, I concluded that it can be an reference list to the LADA Langkawi authorization, and also it could be an further inventarized from the previous study conducted by Dr. Siti Khairia.
Oh, okay, so you have contact LADA before for this result, for these findings?
Sorry, I did not contact LADA.
Oh, okay. Because it is good if you have some results, some findings that you can collaborate with them so that it can contribute something, okay, to their reports or to their for the use of them. Okay, thank you.
Okay, thank you for your
Okay.
recommendation, Doctor. Thank you
Thank
to the panel of
you,
examiners also.
Dr. Siti Nur Asma and Wafika Ismail. Oh, laju ditutup terkejut. Okay, next we have, can I have Nur Azimah? Second
Yes,
last
Doctor.
presenter. Okay. Nur Azimah dengan Dr. Nur Nadia. Alright.
Nampak ke?
Ini screen share kan? Share screen. And then slideshow. Slideshow
Boleh.
mode. Tekan slideshow mode.
Oh, tak gerak
Tak ada lagi.
Dah tekan slideshow.
Dah tekan slideshow. Then maybe you can try to move your slide. Click next. Okay lah, tapi nampak yang slide macam thumbnail kat tepi tu still ada.
Oh, tak nampak Still lagi tak gerak Doktor?
Gerak, dia gerak tapi ada thumbnail tu. Tapi okay kot, nampak je. Okay.
Dah start Assalamualaikum warahmatullahi wabarakatuh. My name is Nur Azmah binti Rohizan and today I would like to present a review on stress in tiger captivity. This is my presentation outline. The tiger, Panthera tigris, is one of the world's biggest cats and is classified as endangered species by the International Union for Conservation of Nature. Tiger
Tak move ke slide? Dah move slide ke? Oh, belum lagi?
Dah.
Oh, nampak Dr. Faiza?
Move introduction kan sekarang? Yes.
Tapi baru je moving kan, tadi tak.
Silah.
Awak tak tekan kot tadi Azimah.
Tadi saya tekan.
Okay, teruskan.
Tak apalah, saya buat macam ni. Okay. Tiger considered as a flagship species for wildlife conservation because of their charismatic nature and alluring features. However, due to the presence of a human, which are human disturbances causing physiological stress. Stress is referred as physiological and neurophysiological alterations in an organism. There are three sorts of stress that my animal might face, which are physical, physiological, and behavioral. Recently, there are a lot of contagion news about the extinction of species in our country, especially Malayan tiger, which only live in the range of fewer than 200 individuals. Due to the current situation, they are expected to have no Malayan tigers left by 2022. Therefore, this study will be beneficial to the zookeepers or wildlife housekeepers in treating their captive tiger. But the most beneficial is to the tigers as endangered species to remain healthy and prevent extinction. The objective for this study are to study what are the stress indicators that used in tiger captivity, followed by determine which is the best stress indicator. The research question for this study are what are the stress indicators that used to examine the stress level? Second, what are the stereotypic behavior that indicated to the stress? Third, what are the factors that affect behavior? Why fecal collection was preferred better than others? And lastly, what are the additional tools other than behavior and hormone? Assessment of stress by behavior. Due to inadequate welfare in animal captivity, animals display abnormal behavior which referred as stereotypes. They are characterized as unchanging and repeated behavioral patterns with no apparent goal or feature. There are some stress behavior which are pacing, auto-grooming, and aggression behavior that display by the tiger. Pacing, which is defined as repeating a defined path around the enclosure at least 2 times. It is shown small exhibitions have a high stereotypical activity compared to the big enclosure. This is because big enclosure need more nutrient. Hence, the tiger in big enclosure could have more happy experience. However, a past study found the overall cage size is not significant with the activity pacing, since it is concluded that the percentage of scan spent on the edges of the enclosure had a strong association with percentage of scan spent pacing. Since the study is long overdue, there might be some inaccurate data going on in this study. Stress also display auto-grooming, scratching, and shaking. This indicator can also be used in wildcat since the rates of these patterns were higher related to the temporary pain or fur abnormalities. However, the findings are not expected since male tiger auto-groom the most in the pre-session of study. which are appear to be utilized as a kind of self-soothing migratory. The tiger may be groomed when they are more relaxed. Moreover, allo-grooming in pair activities couple was discovered too due to the higher stress levels generated by the presence of other tiger. Since stress itself might manifest in form of aggression behavior like striking with paws, biting, pouncing, and frightening other tigers or animal keepers, their study reported increase in restlessness, pacing, and aggression directed at the animal keeper with the feeding cart. Similar observation also was discovered with the most common aggression behavior, which are strike with paws. However, the vocalization and spraying were found to have a positive relationship with this aggression behavior in this study. Next is physiological stress by hormone. Besides stress, hormone is part of physiological parameter that can be used to evaluate the welfare of animal. The release of the glucocorticoid regulates the animal's metabolic status, which is part of the stress response. Stress response was flight-fight response by releasing of catecholamine from the adrenal gland. The study investigates the physiological stress response by using FGCM concentration with resulting in increased FGCM concentration in female tigers linked to the female reproductive hormone cycles. On the other hand, the significant correlation between levels of stereotypic behavior and FGCM was discovered since large rise in FGCM levels seen among tigers kept in smaller cages. In contrast, the varying extraction methods that wet methanol, dry methanol, and dry ethanol were used to assess FGCM concentration and give the high concentration in methanol as a result. The concentration of glucocorticoids can be measured by a few methods, such as fecal, blood, urine, saliva, and hair. However, the fecal method is now widely accepted as the best way since it is non-invasive method. Therefore, the tiger will not be disturbed during the sample collection. In addition, gut microbiota also can considered as possible indicator and play a vital part in the host health maintenance. The makeup of wildlife gut microbiota may alter in response to anthropogenic pressure, which could contribute to stress-related mood changes. Recently, there are few studies reported an increase in composition of gut microbiota involving captive tiger due to the anthropogenic disturbance and feeding habit of the tiger, which involve diet of carbohydrate metabolism, membrane transport, and amino acid metabolism based on the KAGG pathway. Because the intestinal microbiota plays such an essential part in tiger's health, it is linked to the stress. As a conclusion, a few major stress indicators such as stereotype behavior, fecal concentration, and composition of gut microbiota can be used to determine the stress in captive tiger. Most of study largely use FGCM concentration and behavior. However, the best way to avoid inaccurate data is to create variation of indicators. In addition, the community of gut microbiota that linked to a range of stress-related illness, including anxiety and depression in the captive tiger, was recently considered as potential stress indicator. Since it has been involved in wildlife conservation, the wildlife and zoo play a critical role in protection of this ecosystem space. Tigers require large enclosure with physiological enrichment like a water pool, since it can decrease the stereotyping pacing. Therefore, the management should take care of the tiger's husbandry environment to make sure they feel comfortable in the enclosure. Anthropogenic disturbance also will reduce when the number of visitors is controlled, especially during the tourism season. Besides, the zookeeper also plays an important part in welfare since they must provide adequate care in captivity. Hence, the diet of captive tiger should be filled with lots of nutrition to maintain the health of the tiger. Followed by the positive keeper attitude is also important since the tiger's behavior can also be influenced. This is my references. That's all from me. Thank you.
Okay, thank you, Nur Azima. Any question from the floor, from the panel?
Hi, Azima.
Hi.
I'm just amazed at the fact that you said by 2022 that the tiger will be extinct, is it?
Yes.
So we are 6 months away from 2022 and I think we still have about 200 of them. So I think that fact is not true. So can you double check for that? And my second question is, what do you think that the stress factor for those tigers that in the wild compared to the inside the cage?
In the wild tiger.
Yeah, because I believe that all the animals, they do facing stress even if they are in the wild. So do you ever know what might be the stress related?
Um, I think one of the stress factor influence the wild tiger maybe in form of their food. In captive, the zookeeper or the housekeeper um, give the food every day, right? But for the wild, they have to go hunting or prey the food by themselves. If they're not find any food, they might be hunger and have stress. Yeah.
Okay, thank you.
Other than food,
Mating maybe?
yes, other than food, perhaps do you have any idea what other stress contributor to the wild tiger?
Their environment. In the captive, they are located in enclosure, but in the wild, they don't have any territory or play a comfortable territory. So that is maybe one of the factors that influence the stress in the wild tiger, I guess.
And have you managed to answer all your research question in this study as stated in slide number 7?
Yes.
You have
I did.
5 research question.
Yes.
So you can, so you already answered all your research question?
Yes.
Okay. So I think tu saja ke? Ada tak lain? Tak.
Azima,
Oh, okay.
is there any significant difference of the stress level between the male and female
Yes.
tiger?
Yes. There are huge difference between female and male tigers. Especially the females have a high GCM concentration response to stress because of their reproductive cycle. Therefore, it will affect their reproductive to give birth. Give birth to their offspring. To give birth. So there are a huge differences between male and female tigers due to the stress response.
So the highest with the female
Female,
one.
yes.
So instead you mean that female needs more food?
Mm-hmm.
No. The meaning for your answer.
Indirectly.
Indirectly, do female needs more food than male tiger?
Yes, because they need more energy and to carry their cattle.
More
I think
energy to apa tu? To combat fighter ke?
no. I mean some female and male they only have one, but female with carrying their cup. I think this is the word. Anak.
No. Cup.
Cup. Yeah, the cup.
But the subject of your study, do they give birth?
No. Ada saja my study, no.
So you can't conclude based on giving.
Based on my few, there I have stated in this slide that increase of and concentration in female tigers is increased linked to the female reproductive hormone cycles. That is
Hormone.
Yeah.
So kalau dia period je, dia nak makan banyak lah.
Mengandung.
Okay. Thank you panel for the question and also Azimah for your presentation. So we have come to the last presenter for today, Izzah Salsabila.
How come last lah? Nur Aliana? Mana?
Tak ada. Dia
Okay.
Assalamualaikum warahmatullahi wabarakatuh to all examiners. My name is Nur Aliana binti Muhammad Arifin under supervision Sir Shamsul Bahrain bin Ghulam Ali. For this presentation, I would like to explain about the Anthelmintic drug discovery in human whipworm infection. Trichuriasis is a human whipworm infection caused by a gastrointestinal dwelling nematode, Trichuris sakura. These parasites are regularly prevalent among the tropical and subtropical region and could harm humans worldwide. Over the past century, there has been a dramatic increase in approach to develop a treatment to combat this infection. One of the most significant current discussion that triggered a vast scientific inquiry is the Anthelmintic drug. For this presentation, I would like to explain the factors and mechanisms that influence Anthelmintic drugs modulation. There are 5 research questions regarding to this study. The first one is, what are the classes of Anthelmintic drugs that are widely used to treat trichuriasis? Second, what is the concomitant interaction in the efficacy of single and multiple dose? Third, what is the physical-chemical drug properties that influence the efficacy of Anthelmintic drugs? Next one is, what are the regulations in the mechanism of action and in drug resistance? And the last one is, what is the current approach to combat trichuriasis? The objective of this study is to understand the factors and mechanisms that influence Anthelmintic drugs modulation. I will explain about the characteristic of Trichuris trichura. This parasite significantly found in the temperate and tropical environments. Trichuris trichura has a whip-like structure with a short anterior esophagus and a thick posterior anus. This pathogenic whipworm is a multi-host parasite that can infect a wide range of taxonomic groups of animals including swine, mice, and non-human primates. The pathogenic effect can cause intestinal infection by the invasion of the colon by the adult worm and can cause potential hemorrhage, inflammation, and mucosal bleeding in the intestinal mucosa at the site of attachment. The next slide shows the identifications of commonly used Anthelmintic drugs from the classes of microcyclic and benzimidazole. The broad-spectrum classes of microcyclic can be classified based on their structure, efficacy, and persistency into moxidectin, relatively long, persistent, and high efficacy, abamectin, non-persistent and moderate efficacy, and ivermectin, non-persistent and relatively low efficacy. For benzimidazole, it can be classed into several derivatives including albendazole, long persistent and low efficacy, mebendazole, low persistent and high efficacy, and fenbendazole, high persistent and low efficacy. Next, I will explain the concomitant interactions of Anthelmintic drugs in the efficacy of single and multiple dose. The existing Anthelmintic drugs in single treatment have low efficacy against Trichuris trichura. The strategy of combining chemotherapy with other classes of Anthelmintic drug or the multiple dose treatment proved the effectiveness of significant measure in higher cure rate and reduction of egg quantity. The other factor that influences Anthelmintic drugs modulation is the physico-chemical of drug properties. It can be defined as the ability of a chemical compound to elicit a pharmacological or therapeutic effect that is related to the various physical and chemical properties of the chemical substance. The investigations of drugs modulation in the body can be assessed through the concentrations of the soluble drugs. The concentrations of drugs can be measured in the able muscle, small intestine, mucosa, and the target parasite. The other physicochemical property that influences the Anthelmintic drugs modulation is the stability. The stability of drug is measured by various factors including temperature, ultraviolet light, and pH to conduct the susceptibility and efficiency of Anthelmintic drugs. I will proceed with the general mechanism of actions of Anthelmintic. For benzimidazole, it possesses killing effect on worms in the cellular activity stage and can inhibit the mechanism in the cellular transport and energy metabolism. Whereas for microcyclic, it involves the modulation in blocking of parasite neurosignals. They also have an effect on the reproduction of some parasites by reducing oviposition or generating aberrant oogenesis. Next is the regulations of drug resistance. The Anthelmintic drug is being considered as part of a broader worm control strategy, and multiple drug resistance can develop due to the indiscriminate use of commercial medication that are routinely employed for treatment. The key component of resistance to benzimidazole is anticipated to necessitate alterations to the primary structure of beta-tubulin. which role as the building block of microtubules and stimulated to exert their effect on the parasites by binding to the tubulin protein. For microcirculatory regulation, it reduces sensitivity of pharyngeal functioning as a mechanism for anthelmintic resistance and factor mutations that known to reduce microcirculatory susceptibility. The existing research recognizes the critical role of a plant species, Brucia javanica, that can combat the infections from human whipworm. The theory of this plant species plays a role in immobilizing helminth in reducing the infectivity of in vivo and inhibiting the activity adult and age stage of whipworm. In discussion, microcirculatory class has low efficacy to treat human whipworm infection, and benzimidazole shows only poor to moderate response in the clinical efficacy of drugs. The relationship of treatment regimen of albendazole and ivermectin showed a cure rate of 50% compared to the albendazole treatment alone in 30%. The initial understanding of in vivo pharmacokinetic parameters is crucial to predict human interaction in the general mechanism of anthelmintic drug and in the modulation of drug resistance. It is important to detect the causal genetic difference within gene coding for variation that inhibit the drug from physically interacting with the drug target in the mechanism of drug resistance. It is also critical to understand the parameters in the physicochemical drug properties in order to determine the efficacy of an anthelmintic therapy. Other than that, in efforts to combat parasitism, researchers study many scientific approaches to make anthelmintic drugs in various fields, including herbs that are tremendously increased because of their fewer side effects. In conclusion, the reliance on the anthelmintic drug should be reduced by not using them in too frequent periods, and the mitigation strategies such as drug combination can be employed to protect the susceptibility in the effectiveness of the anthelmintic treatment. Last but not least, anthelmintic drugs led to an intense debate of many researchers, and there has been a surge of investigations on the discovery of anthelmintic drugs worldwide.
Can you see my slide?
You can share.
Assalamualaikum warahmatullahi wabarakatuh, and a very good evening to all the panels and my fellow friends. My name is Iza Salsabila binti Mazlan, and today I will present to you my review articles, a review on species composition of red algae, Rhodophyta in several localities, Melaka, Kedah and Sarawak. For the introduction, seaweed belongs to a group of photoautotrophic, multicellular algae growing in the marine environment. Seaweed is a simple organism that can conduct photosynthesis. According to the study conducted by TAS in 2016, it states that seaweed contribute over 19% of global photosynthesis as a primary producer. East Asian has used seaweed abundantly as a raw material in fruit, medicals, and plantation industry. This is my research question regarding my review articles. How many families present in several localities? What is the numbers of family of Rhodophyta present in Melaka, Kedah and Sarawak? Which localities have the most presented family of Rhodophyta? In that locality, which family of Rhodophyta has the highest number of presented? And lastly, what is the difference between that locality with other localities? For the aim of these review articles aim to analyze, observe and describe the species composition of red algae, Rhodophyta in several localities based on the previous related articles. For the main body, based on pigmentation, seaweed is categorized into three main divisions, Rhodophyta, Chlorophyta and Phaeophyta, and the mostly dominates the marine water is Rhodophyta. Rhodophyta, also known as red algae, is an autotrophic protist that belongs to the kingdom of Archaeplastida, where it is the largest phyla of algae that consists of 6,000 recognized species. Rhodophyta is a red seaweed due to primary pigment, chlorophyll A, chlorophyll B, and phycoerythrin. However, not all Rhodophyta species are red in color. Their pigmentation can be slightly varied depending upon the type of water. A few species of Rhodophyta are known by the capability of living in the deep water. The more profound species in the deep water are typically dark red, slightly bright with more pyrethrin pigment that can absorb blue light wavelength. Moving on to the discussion part, Table 1 shows the checklist of Rhodophyta in Sarawak. There are 12 numbers of family of Rhodophyta that presented in Sarawak. Next, Table 2 and Table 3 show the checklist of Rhodophyta in Kedah and Melaka respectively. There are 15 and 8 numbers of family of Rhodophyta can be seen in Kedah and Melaka. Next, in Table 4 show the numbers of family of Rhodophyta present in several localities. In this table, I have compiled 3 localities, Melaka, Kedah and Sarawak, to observe the highest and the lowest of locality that has the highest and lowest numbers of family of Rhodophyta. As you can see from the table 5, Kedah recorded the highest number of texta compared to other two. This is due to the sampling site conducted in Kedah consists of 20 sampling sites, while in contrast, the sampling site in the Melaka and Sarawak consists of 4 and 6 sampling sites respectively. In Kedah, the highest number of family present is the family of Gracilariae. However, the family of Gracilariae only present in Kedah and Sarawak but absent in Melaka. The type of species can be found is Gracilaria salicornia. Gracilaria salicornia is very successful in calms and protected water, where it can be found in tide pool, on reef flats, and intertidal to subtidal. Gracilaria salicornia was found in the air hangat mangrove, Sungai Klim near the River Mouse in Kedah, and subtidal and intertidal of Tanjung Batu, Kuala Nyalau and Pantai Telekom that is near to the tide pools and reef flats, which are the growing areas of Gracilaria salicornia. Thus, it contributes to the numbers of family of Gracilariae. Meanwhile, in the Sembilan group, when the sampling was conducted, there was a strong wave in Pulau Lalang and Pulau Rumbia. Since Gracilaria salicornia is very successful in calm water, hence there are no appearance of this species. Moving on, in contrast, in Melaka, Melaka has the highest family of Rhodomelaceae, followed by Sarawak and lastly Kedah. Both Melaka and Sarawak has the similar species found. However, this type of species is absent in Kedah. The name of the species is Lawrencea species. Lawrencea species are mainly found in warmer water, where the optimum temperature growth is 25 degrees Celsius. When the sampling is conducted in Sarawak, it's mainly focused on intertidal and subtidal zone, where the range of temperature is between 23 to 38 degrees Celsius. Therefore, it contributes to the abundance and growth of Lawrencea species in that area. In addition, Lawrencea species can grow in various habitats via tide pools, rock, and corals. In Melaka, top species of Lawrencea species are found in coral, where the species is growing. However, Lawrencea species could not be found in Kedah, where the temperature of Sungai Kelim Kedah is 27.1 degrees Celsius. Sungai Kelim Kedah recorded the highest numbers of family of Rhodomelaceae in Kedah. So the primary factor of Sungai Kelim, that the temperature is higher from the optimum growth temperature for Lawrencea, is not suitable for the growth of Lawrencea species. Next, the family of Helmintiaceae were present in Sarawak and Kedah. The species found in both localities are Helmintia dilatata, Helmintia maculata, and Helmintia durvillei. Here's the figure for this species. These species have the characteristic that they're very slimy when fresh. And these species are commonly found attached to rock in the lower intertidal to upper subtidal area and moderately exposed to wave action. However, according to the previous study, the numbers of Helmintia are seen to decrease. This is due in island in Kedah had a high cover of barnacles and rocky oyster that attached to rocky sides. Hence, Heliminia had to compete for space and appear to lose to the barnacles. In the meantime, in Sarawak, the numbers of Heliminia might be affected in a seasonal pattern due to their very slimy character, causing disability of the species to survive in wet season because of a high degree of wave. This can be seen in Sembilan group, where there are no appearance of the family of Helimiaceae due to heavy rain causing strong wave, where the sampling is conducted. In conclusion, the highest numbers family of Rhodophyta recorded is in Kedah, where 15 families has presented during the expedition, and the family of Rhodomelaceae documented the highest with the total of 32 species in 3 localities. For the suggestion, I suggest a future study about the species composition of Rhodophyta more extensively to magnify the division and comprehension of red algae composition. And more study on composition of Rhodophyta can be done with better time span of study after this pandemic so that every family collected can be reported. That is all from me. Thank you.
Okay, thank you, Iza. Any question?
Iza,
Yeah.
I just want to ask, is the red algae being harvested for any economical value?
Harvested. Yes. For the example, as I have mentioned before, the Heliminia, the numbers of Heliminia were decreased from time to time. This is due to seasonal factor or this is due to seasonal pattern. Other than that, there are species that if I'm not mistaken, are gracilaria that the numbers is decreased due to environmental factor such as the changes in ocean salinity and light intensity. So it directly changed the numbers of seaweed found.
Okay, thank you.
Thank you.
Any other question?
Iza.
Yes.
At the first place, why you are choosing Melaka, Kedah, and Sarawak? Yeah.
Because I have go through all the research related with rhodophyte related with seaweed, and Melaka, Kedah, and Sarawak has Melaka, Kedah, and Sarawak the result from Melaka, Kedah, and Sarawak shows the Rhodophyta as the highest numbers. So that's the main reason I choose that localities.
Oh, okay. How about Sabah? Yeah. Do you have any idea?
Sabah, I have go through the from Sabah, but I think because I don't.
Yeah, because Sabah import the highest import seaweed from Sabah, if I'm not mistaken. That's why I just asked if you have any idea on Sabah production of seaweed.
Sorry, I don't have any idea.
It's okay.
Okay. I think maybe one last question from me. How can you see this is your article review, right?
Yes.
Okay, so after you have read all the Rhodophyta, is it Rhodophyta?
Yes, Rhodophyta.
And Rhodophyta. So what can you suggest in terms of any potential study that might that perhaps can increase the number of Rhodophyta in the selected state? Maybe like not Melaka lah. Apa lagi dua tu yang like
Sarawak and
Sarawak and Kedah.
Okay, for me, I think because the numbers of few species from Rhodophyta has shown the decreasing. So I think the best thing that we can do is by the authorities to build a, to build a place or something that to secure the production of Rhodophyta in the future. Because I think if we are not doing this now, I think we will the numbers of Rhodophyta will decrease from time to time.
So did you put the suggestion in your article review, or this is just your saying?
This is just my saying.
Oh, okay. Okay, thank you. Perhaps no other question, yeah, from the panels. And this is our last presenter. Last presenter. We have only 10, 10 candidates for today. So Alhamdulillah, we have come to the end of the session. Thank you to all the students for great presentation. And also because you guys remain stay until the last presenter, and not to forget also many thanks to the lecturers for your time and effort in evaluating the session as well. So I guess all the important deadline that the student must follow, and also the things to do right after presentation, macam tarikh kena hantar and everything, it was already mentioned by coordinator Dr. Wan Roziano in the work schedule kan. So I just leave it to you guys to check back your deadline, and maybe you can also tally with your supervisor. Perhaps I'm not sure, maybe nak return the correction ke. And I rasa itu sahaja kot untuk sesi hari ni. Okay kan? Tak ada apa-apa lagi kot kan? Dr. Siti Khairia, Dr. Siti Nur Asma, Dr. Nadia.
Tiada.
Kan? Okay kan?
Dah habis.
Alright. So till then, Assalamualaikum. Stay safe semua and stay healthy.
Okay, thank you.
Thank you.
Pengerusi tak ada minum-minum kan? Minum petang.
Ada. Nah, nanti tak ada lah.
Bye semua orang. Semua buat report.
All the best. Congrats.
Thank you, Doctor.
Okay.
Thank you, Doktor.
Tahniah, settle habis satu kerja yang leganya. Aku yang lega, Kak.
Alhamdulillah.
Saya pun
lega kan? Satu benda.
Diorang lah kena struggle ni buat writing kan lepas ni, buat pembetulan.
Okay.
Selamat berfasa bertenang ya.
Jangan
Tak
stress
bertenang
sangat,
juga, Doktor.
Bertenang sangat.
Tak bertenang juga rasanya.
Fasa bertenang korang tu. Korang dapat seminggu apa? Macam mana?
Tapi I rasa diorang dapat 18 kan? Sampai 18 Julai.
18 Ahad kan? So sampai Ahad je ke?
Tak tahulah. I think so.
Tempoh bertenang daripada 16 sampai 21.
Apa masa fasa bertenang tu?
21 to
bertenang.
21.
Kiranya
Tak boleh ada apa-apa submission.
tak ada submission.
Dari
assessment.
Again.
Tak boleh ada apa-apa submission, test, assignment, anything.
Daripada?
I think 16 until 21.
16 until 21. Okay, so you
ni
punya
yang fasa
submission.
bertenang student, fasa bertenang aku mana? Okay.
Only 2 week, study week tu.
Betul, study week. So maknanya you all punya thesis ni kena submit bila ni? Thesis ke manuskrip ke apa benda tu kena submit bila? Dah kalau tak boleh submit.
Dr. Wan belum update lagi.
Oh, okay.
Dr. Wan cakap dalam grup sebelah tak ada perubahan.
Kan? Betul.
Itu yang I macam confused. What is fasa bertenang?
I pun baru tahu ni bila
Tapi
Kai cakap.
Lilin ni baru keluar.
Baru ya.
Teknologi industri baru dapat je. Ni ada petition baru, tak boleh ada any submission of assignment ke quiz ke apa pun presentation ke during that period.
Daripada bila kakak ulang balik pada Sabtu ni?
16 hingga 22.
16 Jumaat. Okay, so dia kena hantar Khamis lah macam ni.
16 hingga 21 kan Akasha?
Ya.
Sebab 22 dah start final test.
So kita hantar awal lagi lah korang. Jom kita hantar awal.
Okay korang, bye-bye.
Thank you,
Assalamualaikum.
Thank you, Doktor. Kita ni last jumpa, dah tak jumpa dah.
Alah sedihnya dengar. Okay, bye. Assalamualaikum.
Salam, bye.
Thank you, Doktor.